let's talk about transcription. And when we talk about transcription, we have to start with the central enzyme that carries out transcription. That's the RNA polymerase. So what kind of RNA polymerases do we know? When we look at RNA polymerases in cells, we see that bacteria and archaea have only a single enzyme, whereas eukaryotes have 3 RNA polymerases, 1, 2, and 3. And you even find 2 more polymerases in plants, Pol4 and Pol5, which are actually most similar to RNA polymerase II. Now, what's also important to realize is that in eukaryotes, these different polymerases transcribe different classes of genes. Pol1 mainly makes the ribosomal RNA, Pol2 the messenger RNA, but also some non-coding RNAs. And then Pol3 transfer RNAs, but also other important non-coding RNAs. A little bit of history is important. And that is how was it discovered that there's 3 RNA polymerases in eukaryotic cells. Now you are doing your master and soon you will do a PhD. You will go to the lab and hopefully you find something very exciting. You have to be prepared for that. That's very important. This is called serendipity, right? It's not just plain luck. It's also that it will hit the prepared mind, right? So be prepared for finding something very important. And there was a graduate student over 50 years ago in the US who one night got this chromatogram. At that time, of course, you would draw with a pen on this kind of paper. And he saw 3 peaks, 1, 2, 3. That's a salt gradient. So there were 3 activities eluting. And at that time, of course, you would measure activity. So there were 3 activities that could produce RNA, and that was Bob Roeder. And here he is. That is now 3 years ago, 2019, because we celebrated the 50th anniversary of his discovery of the 3 eukaryotic RNA polymerases at Cold Spring Harbor. And we put the 3 enzymes, the structures of the 3 enzymes on the cover of the abstract book. And he gave this, beautiful evening lecture on the first day. Everybody, applauding, standing up, standing ovations, celebrating the discovery, because really he is the founder of that field by, as a graduate student, by detecting these 3 peaks of activity. And that is why the enzymes are called Pol1, Pol2, and Pol3, because simply it was the first, second, and third peak on the column that Bob detected when he was a PhD student. Now, why are these enzymes so important? If you think about making protein, you really have to think about all 3 polymerases, because Pol1 makes ribosomal RNA that is essential to make a ribosome. And then the tRNAs are products of Pol3. They will fuel the ribosome, right, to bring in the amino acids. And the messenger RNA, of course, is the message that instructs the ribosome on which protein sequence to synthesize. And we have now all 3 structures of the enzymes, so we can compare them. And that is, of course, a starting point for analyzing how transcription works in mechanistic detail. But before we look into that, let's look at the activity of the 3 RNA polymerases in cells. Here on the left, you see the number of genes that each of those polymerases are transcribing in yeast cells. And you see the vast majority of genes, over 90%, are transcribed by RNA polymerase II. It transcribes all the protein coding genes. And then Pol1 and Pol3 only transcribe a small fraction of genes. But when you look at transcriptional activity in cells, the picture is entirely different. Now suddenly Pol1 shows 60% of the activity of making RNA, right? It's really the workhorse of the cell, making ribosomal RNA. And Pol2 only a smaller fraction, and Pol3 also a small fraction. And then finally, when you look at, you purify RNA from yeast cells and you look at the amount of RNA you find in cells, you see that 80% of that RNA is actually ribosomal RNA. So it's all made by Pol I. And a large fraction, 15%, is also made by Pol III, all the transfer RNAs and other non-coding RNAs. And only a tiny fraction is made by RNA polymerase II, because the messenger RNAs are not made at large quantities, high levels, only few copies. And why is that? It's because you can reuse it, right? You can use the same messenger RNA molecule multiple times to synthesize a protein. Now, I've shown you in the little introduction to structural biology this reconstruction of polymerases making ribosomal RNA. And this has actually been seen already 50 years ago. These are the so-called Miller spreads. So you can purify these loci, the ribosomal RNA encoding loci from cells, and you can then see, this is the DNA thread here, you can see these RNA polymerase I enzymes trailing down the DNA, and as they transcribe the DNA, they make RNA molecules. These are the little extensions from the DNA. And as the Pol I enzymes move down here, the RNA product gets longer and longer and longer. And this is why you generate these kind of Christmas tree structures. You can even see at the end of the RNAs these little knobs. And these knobs are ribosomal RNAs that are beginning to fold, ribosomal subunits that are beginning to be formed at the end of the RNA product. Now let's zoom in. You see here these black dots along the DNA. These are the individual RNA polymerase I enzymes. And Christoph used to be a PhD student in the lab, and he was really the one who solved the RNA polymerase I structure. It's almost 10 years ago. And just to give you an idea how it worked, and it will still be similar today despite all the machine learning and so forth, it needs human intervention if you want to solve a very, very complex structure. And the way he did it, he put all the sequences of the 14 subunits, amino acid sequences, on the wall, and he tried to fit those sequences into the structure using 3D graphics. You have to find the right place where to fit it, and you have to build the atomic model. It will be easier nowadays due to all the methods that I described before the break, but still it needs this human intervention to make sure you have the right structural model at the end. And this is then how the model looked like. So you see here all the atoms as little balls. This is RNA polymerase I. And the different colors represent the different subunits of the enzyme. You see this major cleft here running through the center of the polymerase. And you also see how the subunits interact. It's actually a very complicated enzyme where these different protein subunits have co-evolved over time to give the polymerase various functions. Also a historic slide by now. This was my own work when I was still a postdoc at California with Roger Kornberg at Stanford University and solved the first structure of such a eukaryotic polymerase, RNA polymerase II. And it was so exciting when we first got the structure, because of course we wanted to know, where are the conserved regions? And we saw here in orange, the conserved regions are all around the active site, which is symbolized here with a sphere, which is what is called the metal ion A. It's the catalytic site of the enzyme. And then we plotted, for the first time, the distribution of charges on the surface. And we got this really drastic image where the inside of the enzyme is positively charged, the outside is negatively charged. Obviously, it needs to attract the negatively charged DNA in the active center cleft. And then we zoomed into the active site and we wanted to know how does it really look like at the catalytic site. And we saw this loop here, which is called the aspartate loop. It has 3 aspartate residues. And those 3 aspartate residues hold the metal ion A. You see here what is called an anomalous difference Fourier map, which can give you, by X-ray diffraction, the exact location of the catalytic metal ion. It's like a jewel in a crown, you know, and the crown is formed by these aspartate residues. And this motif actually you find in all living cells, you find in any bacterial polymerase, archaea, pol1, pol2, pol3, pol4, pol5. All these enzymes always have this loop and the same catalytic site. So there's a single ancestral enzyme from which all these polymerases have emerged. So we know now also the mechanism of catalysis, and that involves not only one metal ion, it actually involves a second metal ion. And this second metal ion, or metal B, is recruited ad hoc, so it enters together with the nucleoside triphosphate substrate, and then the 2 metal ions are both in the active site and catalysis can occur. And you all know how it works. You have here an OH group that is attacking the alpha phosphate of the incoming nucleoside triphosphate. And this 3' OH group is the growing end of the RNA in the active site. And the whole reaction is what is called an SN2 type of reaction. It's a nucleophilic attack of type 2. So you attack this alpha phosphate, and pyrophosphate, these 2 phosphates together, is cleaved off and is leaving the enzyme, most likely together with metal B, and then you finish this cycle, which is called the nucleotide addition cycle. Now, that much to the basics of the polymerase. We will discuss more the reaction of RNA chain extension next time when we discuss the elongation phase. But before we can do that, we need to first understand how initiation works, right? We need to first understand how the polymerase actually finds its promoter, how it opens the DNA, and how it starts RNA synthesis, how it initiates the formation of an RNA chain. So this is called initiation, the initiation of the RNA chain, but it's much more than that. As I said, it needs to find the point where it starts. It needs to bind to the promoter, open the DNA, and then start synthesis of the RNA chain. So how does that work? Let's go and talk about transcription initiation. Before we do that, we need to define a few elements so that nobody gets confused here. Of course, everybody would agree that there has to be a transcription start site. This is actually the nucleotide where RNA chain synthesis initiates. And this will later be the end of the RNA chain, right? When you have a complete messenger RNA chain. The polymerase will then move all the way to the so-called polyadenylation site, and it will even run over the polyadenylation site until it reaches a transcription termination site, right? But at the poly A site, the transcript will be cleaved and a poly A tail will be added. So the final mRNA transcript will run from here to the poly A, and then there's a poly A tail added to it. Now, upstream and downstream is what we call the regions that flank the protein coding region to the upstream and downstream side, and the directionality is given by the movement of the polymerase. So the movement is from upstream to downstream. This is how we define it. And this whole region here around the transcription start site, it's called the core promoter or the so-called basal promoter. But there's also sites in the DNA that can be either close by or far away that can regulate what's happening at the promoter. And those that are far away are called the enhancer elements. The others are called cis-regulatory proximal elements. So transcription factors can bind there and they can regulate the process of initiation. But the actual transcription initiation machinery will assemble at the promoter around the transcription start site. Now, the general cycle, I think, is clear to everyone. Once you have assembled a pre-initiation complex, you can open the DNA, start forming the RNA chain. Then the polymerase escapes from the promoter into the transcribed region and forms the so-called elongation complex. This is the elongation phase, right? It runs over the polyase site, and then when it reaches the termination site, it will be released from the DNA. And that's called termination. So these phases that you see in every textbook, initiation, elongation, termination, they're actually defined with respect to the RNA chain. You initiate the chain, you elongate the chain, you terminate chain synthesis, right? Always remember that, because it's sometimes not so clearly defined in books. And what is interesting is that it actually needs dozens and dozens of factors, even hundreds of factors in the case of RNA polymerase II, to go through that cycle. Yeah, you may say, why is this so complicated? This, of course, has to do with regulation. It has to do with co-transcriptional processes, because remember, the RNA needs to be kept, spliced, polyadenylated, exported from the nucleus. So all these things happen co-transcriptionally. A lot of factors have to be involved, and it needs to be tightly controlled, because it really matters how many copies of mRNA you make over time. How does the polymerase first get to the promoter, right? Because the DNA is normally packaged into chromatin. So you need to first open chromatin around the promoter. You need to have a situation where the pre-initiation complex can be formed, and it can only be formed on naked DNA. The promoter needs to be accessible. And for that, we have so-called pioneer factors that can bind to nucleosomes, destabilize them, and they can also recruit large complexes, so-called chromatin remodelers. And these remodelers use the energy of ATP hydrolysis to shift around nucleosomes. They can also evict nucleosomes, and basically then you have a free DNA region where you can recruit co-activators such as the SAGA complex and others that can, for example, acetylate chromatin, make it more transparent. But in this open DNA region, the so-called nucleosome depleted region, you can then have an accessible promoter which can allow for the assembly of the pre-initiation complex. So very briefly, I show you a movie of the first pioneer factor structure on a nucleosome to give you an idea how that looks like. You all know the nucleosome structure. And now a pioneer factor, the so-called SOX2 protein, which is a Yamanaka factor, will bind to nucleosomal DNA. And because it clashes with the neighboring DNA gyre, it can destabilize the DNA and unravel parts of the DNA from the nucleosome. Now you see these terminal pieces of DNA, they have been unraveled, and they can now also be bound by transcription factors and other proteins. And what is important to recall is that these pioneer factors not only allow you to enter chromatin because they bind to nucleosomes rather than naked DNA, they also have these very long intrinsically disordered regions, right? And those are basically used to recruit large complexes like these chromatin remodelers that help you to open chromatin. So pioneer factors can first mark the site where you want to open, then they recruit other factors to open. Now you have an open DNA region, a nucleosome depleted region, and now the initiation complex, the pre-initiation complex, can actually assemble. Now this pre-initiation complex for RNA polymerase II contains all these other factors here. TF2A, B, you also have the TADA box binding protein, which is part of this factor, TF2D, TF2E, TF2F, and TF2H, and also another co-activator, the mediator complex. So it's a very large assembly, right? And that is again because you want to regulate initiation very tightly. And it's also to recognize the promoter properly and only start at certain sites of the genome and not at other sites, right? Otherwise you make the wrong RNA products. When you look at archaea, actually it's much simpler, but you have a subset of those factors that are actually homologous to some of the eukaryotic factors, Tata box binding protein TBP. Then the TF2B has a homolog which is called TFB, and TF2E has a homolog called here TFE. So it's like a minimalist version of the eukaryotic complex. But in bacteria, it's very different. You have the sigma factor that binds here to promoters. It's actually the protein that is seen here in magenta. And it's basically a single protein. You can have different types of sigma factors for different types of promoters, but it's much less complicated. So it's the result of convergent evolution. The polymerases have the same origin, but these initiation factors have been derived later, and they are unrelated between bacteria and then archaea eukaryotes on the other side. So we look now at this complicated process of RNA polymerase II transcription, and we start with simple promoters. We start with promoters that have a Tata box, and this Tata box is a sequence element that is located upstream of the transcription start site, about 30 to 40 base pairs upstream. Interestingly, only a fraction of promoters has a Tata box. And I will come back to the question, how does it work at other promoters when there's no Tata box, right? But let's start with something simple where you have a Tata box. You can have then the Tata box binding protein which binds to the Tata box, and this can recruit factors TF2A and TF2B. They will bind to flanking sites, right? And this complex is called the upstream complex. It's upstream of the transcription start site. Now the promoter is marked and it can actually recruit RNA polymerase II. But it can only do that when the polymerase also is bound to a factor called TF2F. Then you can form this assembly where now the polymerase is positioned around the transcription start site. Now you still need two other factors, TF2E and TF2H. And actually, these are both multi-subunit factors. TF2H actually has 10 subunits. Pol2 has 12 subunits. So the whole complex here, together with the mediator, coactivator, is at around 50 or more subunits. It's a very large assembly. So what is the mediator doing? It actually, in vitro, can stabilize the situation. In vivo, it looks more like it helps the PIC to form. It's almost like an assembly factor that helps the pre-initiation complex to form. And then DNA can open, but the polymerase will not escape from the promoter yet. For that, you need to phosphorylate a part of the polymerase that is called the C-terminal domain, or CTD. This is a long, intrinsically disordered tail-like extension. And there's a factor within TF2H, a kinase called CDK7, which can phosphorylate the CTD. And when the polymerase is phosphorylated, and also other factors are phosphorylated, the whole pre-initiation complex is destabilized, and now the polymerase can escape from the promoter. And the whole process can be regulated by activators. And what do these activators do? Well, they often recruit factors that are needed for initiation. In particular, this co-activator, the mediator complex, but also others like acetyltransferases and so forth. Now, what do you estimate? How many of these transcription factors do you actually have in human cells? Do you have any idea of these activators? These general factors are, of course, the same at all genes. You will use the same factors on all genes. But these activators will be different for different classes of genes, right, that you want to activate. You have any idea how many such factors are encoded in our genome? 10, 100, 1,000, 10,000. Yeah, 1,000 is a good estimate. It's almost 2,000. So we think about 8% of our genes encode for transcription factors. They all have a DNA binding domain, and then they have usually these transactivation domains that are also disordered regions that can bind to co-activators and other factors. And this way they can stimulate transcription initiation from specific genes because they will only bind to their cognate DNA site, right? This is how you make sure initiation occurs only at a subset of genes, because obviously not all the 22,000 genes are active in all cells, but only a small subset, maybe a few thousand maximum. And that defines which cell type you have, and that defines also the functions, which proteins are made in the cell. Now here's a movie based on the structure of the pre-initiation complex, of how in a stepwise manner such a pre-initiation complex may assemble. You start with the TADA box binding protein in red. The DNA is bent by 90 degrees, and then TF2A in yellow and TF2B in green are bound. And now the polymerase is recruited mainly through an interaction with this domain here in TF2B, but it needs TF2F, which is this pink factor on the side. The promoter can then be retained because TF2E and TF2F, they bind to each other on top of promoter DNA. So now basically these factors encircle the promoter DNA, they retain the promoter. Now TF2H can bind, which is this large 10 subunit factor. It binds downstream. There's a minor movement in the DNA when it binds. And now this is what we call the pre-initiation complex. But as I said, mediator is important to steer the whole process, also to stabilize the pre-initiation complex. And mediator has also this kinase that is called CDK7 within a kinase module. And that is so important for phosphorylating the CTD of POL2 to trigger the escape of polymerase from the promoter. Now we have now structures both of the yeast and the human transcription pre-initiation complexes, shown here on the left and on the right respectively. And the structures are incredibly similar. This shows you how well conserved these structures are. But why is it important to work on different systems? It's, for technical reasons. You try to analyze certain aspects of transcription, and you can do it sometimes in the yeast system, sometimes in the human system. I will show you one example. We wanted to understand how does the polymerase open the DNA, right? How does that work? And we could only understand it by studying both the yeast and the human system, then bringing the evidence together. So in the yeast system, we could stabilize a very early intermediate of DNA opening. You will see that in a movie here. Basically, we just see the beginning of DNA opening when torsion is introduced. So here's TF2H, and there's an ATPase here in TF2H, a translocase that hydrolyzes ATP to produce this mechanical strain in the DNA. So here is the translocase, and now it's propelling the DNA into the active site. And you see that these base pairs here are beginning to be loosened. It's a very early intermediate with a tiny little transcription bubble being, at the beginning of the transcription bubble being formed. And this is stabilized by neighboring elements in polymerase and TF2F. So this we could visualize in the yeast system. But then in the human system, we could actually visualize how that translocase works. So now you see only the translocase. Everything else is removed so that it's simple. And you see this ATPase, this translocase, it binds to the DNA, it introduces a distortion. You see this distortion here. And then when it hydrolyzes ATP and there's a conformational change, you see this twisting motion, and actually the DNA is translocated by one base pair in the upstream direction. And this is basically the twist that causes the beginning of the opening of the DNA, because there's now strain being introduced into the DNA, and the two DNA strands begin to separate. Now, we don't know for sure how often this translocase needs to push the DNA by one more base pair, maybe a few times. We're not sure here, but this animation shows you how it works. So it's, if it would work multiple times on the DNA, it would propel the DNA into the active center of the enzyme. It's possible that you need to do that only a few times, and then the polymerase can suck in the DNA because it has this positively charged cleft, remember? So when you begin to open, then maybe it's very unstable and the rest of the opening happens spontaneously. So maybe you don't need to do that 10, 15 times, maybe 1, 2, 3 times, 1, 2, 3 cycles of translocation are enough. But then you form here a complete DNA bubble. And now the DNA is reaching, the active site of the enzyme. It's not, in the beginning, it's suspended 40 angstrom above the active site. But then when you melt the DNA, when you open the DNA, you form single strands. It becomes very flexible. Then the DNA can enter the active site, and it's moved by 40 angstrom. And the DNA template strand is actually inserted into the active center cleft. And now it's running past the active site of the polymerase. And then you can actually initiate RNA chain formation, because now you have a DNA template in the active site of polymerase. This is just a schematic where we've cut through the structure. You see here the active site of the polymerase. This is the ATPase, which is propelling and pumping the DNA into the active site during the opening. And now the blue strand is the DNA template strand, and it's now running past the active site of the polymerase. The cyan strand here is the non-template strand. So the template, of course, is used as a template, as the name says, to direct RNA synthesis, which NTP is selected and which nucleotide is incorporated into the growing RNA chain, whereas the non-template is just running on the outside of the active site and then forming a duplex again beyond the active site on the upstream DNA. Now, when we look in cells and ask the question, do all promoters need this ATPase, or can some of them maybe open DNA spontaneously, as happens in archaea, as happens in bacteria, as happens for POL1 and POL3, then the answer is yes. There's also POL2 promoters that can open spontaneously that don't need this complicated mechanism. So what we did here is we used these cells, and we used a method where you can very rapidly remove the translocase that we just looked at from the nucleus. And then we watch RNA synthesis in the cells using RNA labeling techniques and RNA sequencing. And then what you see is that about 80% or so of the genes rapidly lose RNA synthesis activity. That means they really need this ATPase. They follow that mechanism that we just looked at. But there's also maybe 15 or 20% of the genes that are still fine. RNA is still synthesized. Also, there's no ATPase in the nucleus. So when you then look at the sequence of those transcription start sites and those promoters, you see that the initially melted region of the DNA is actually evolved to have a sequence that gives an unstable DNA duplex. So it's a region that evolved to be easily opened, where the two DNA strands are easily separated. So it's another level of regulation, right? You can have promoters that don't need that mechanism, but the majority of promoters need it. Now we come to this interesting question, when most of the genes don't have a Tata box. Yeah? So between 80 and 90% of the genes don't even have a Tata box. Right? And what I showed you, of course, relied on the Tata box because you initiate pre-initiation complex assembly at the Tata box by the TBP protein binding there. So how can all those genes that don't have a Tata box, how can they know that, or how does the polymerase know where the beginning of that gene is, where the promoter is, where the transcription start site is? So where should the pre-initiation complex assemble, right? How is the beginning of the gene defined? Now, this is still ongoing research, but I can show you two examples here that point in the direction. So one thing that seems to be important for directing the polymerase to the start of the gene is the +1 nucleosome. Remember I told you that when you form this nucleosome depleted region around the promoter, there's nucleosomes, some may be evicted, but then nucleosomes are also shifted to the side. And then you have this DNA region that is nucleosome depleted where the pre-initiation complex assembles. But now the +1 nucleosome is the first nucleosome in the gene that the polymerase will encounter. It's this nucleosome here. And this is not published yet, but we know that the pre-initiation complex can assemble just next to it. So the idea is the following. The cells are forming these nucleosome depleted regions, and the +1 nucleosome will end up at a certain site. And then the pre-initiation complex assembles on the naked DNA, and because there's a +1 nucleosome, it can use that as a border element to define its location, its position. That is the idea. And that would actually explain, why initiation occurs at all these other genes at a certain site, at a certain distance also from the +1 nucleosome. On the right, you see something very different. This is now for Pol2 genes that don't encode for a protein, where not messenger RNA is a product, but a non-coding RNA is formed, for example, snRNA. And so at these promoters, it's again different. They don't have a TADA box, but instead of a TADA box, they have an upstream sequence element that binds a different factor called SNAP-C. So here you have an additional factor that tells the polymerase where the pre-initiation complex should assemble. And this SNAP-C factor then binds, TF2A, TF2B, forms this upstream complex, and that can then recruit polymerase together with TF2F. So there's different solutions. There's not just the one solution I showed you in the movies, but there's these alternative solutions how the pre-initiation complex knows where it should sit on the genome. Now we briefly compare, just a second, the pre-initiation complex of RNA polymerase II to those of RNA polymerases 1 and 3. And you see the Pol2 complex is larger. That has to do with regulation, but it's also larger because it has this fancy mechanism of DNA opening with the translocase. And you don't need that in the case of Pol1 and Pol3. They both open the DNA spontaneously, so the pre-initiation complexes are also smaller. You had a question, please.
I saw there was TBP in the previous slide.
Yeah.
In the non-coding RNA promoter.
Absolutely.
So why is it present?
Yeah, that's a good point. Okay. So basically, TBP binds, also there's no TADA box. And that's the same, in these non-coding RNA promoters, but also at such promoters that don't have a TADA box. So TBP can bind non-specifically, and it will still bend the DNA. The bend will probably not be 90 or 100 degrees, it will be a little bit less, maybe 70 degrees, 80. And it will probably not sit, as closely to the DNA bases as it does in the promoters that have a TADA box, but TBP is always there. And, it's basically due to all these protein contacts around it with TF2A, TF2B. Yeah. So you see that here also, in the case of Pol3, you have again TBP, but it has a different set of factors around TBP. So it really seems to be a universal factor in eukaryotes, also in archaea. Now, next time, I want to take you from initiation to elongation. Here you see an elongating polymerase. Now the polymerase has escaped from the promoter. It formed this elongation complex. And you see in red the RNA product that has been formed. And now you have a different set of factors. Two factors are actually shown here. These are now the elongation factors. So basically what needs to happen when you go from initiation to elongation is that, of course, you start RNA synthesis, you extend the RNA chain, but also you exchange all the protein factors. You have to get rid of all the initiation factors, and then the polymerase needs to assemble with the elongation factors, right? And so the initiation factors can then be recycled for recruiting the next polymerase to the promoter and helping the next polymerase to open DNA and to get started. And with respect to the elongation factors, you know, we only learned over the last years that there's so many elongation factors that travel with polymerase and that are needed for all kinds of processes. They are needed, of course, you know, to make the polymerase processive. So for example, you know, the average human gene takes 40 minutes to be transcribed. So these genes are extremely long because they have very long introns. Imagine if the polymerase would fall down the gene after 30 minutes. You would lose, you know, all that transcript. So that it needs to be processive, and there's factors that render the polymerase more processive. Processivity means that the process gets completed. It's not interrupted by early termination. That's why you need elongation factors. Second point, you need to stimulate the catalytic process at the active site of the polymerase. So there's actually elongation factors that either directly or indirectly can improve the catalytic efficiency of the polymerase. The polymerase now suddenly is faster, and there's also elongation factors that make the polymerase accurate. So it has an intrinsic accuracy, you know, it makes few mistakes, but sometimes it incorporates the wrong nucleotide. And then you need a factor that is proofreading the RNA, that is actually correcting that error that has been made. It's also a class of elongation factors. Then you also need elongation factors to help the polymerase to go through chromatin, right? There's all the nucleosomes now in the way. And imagine at the speed that we see in living cells, the polymerase has to go through 10, maybe 15 nucleosomes every minute because it transcribes 3,000, 4,000 base pairs per minute. This is how fast it is. So you need elongation factors not only to help the polymerase go through nucleosomes, but also to prevent a situation where all the histones are lost, right? Because if you would lose the histones while the polymerase is going through the gene, you would also lose the information that is stored by modifications of the histone tails, you know, acetylation, methylation, and so forth. So you have also factors that travel with polymerase that prevent the loss of histones and help the nucleosome to be reassembled in the wake of polymerase once the polymerase goes through. And you have elongation factors that help the polymerase to modify the RNA. This is, of course, done by additional enzymes. Capping enzymes, the spliceosome, the 3' processing machinery, right, which has an endonuclease to cleave the RNA. It has a poly-A polymerase to make the poly-A tail. And all those factors are recruited to the transcribing polymerase, to this complex, through additional elongation factors. So this kind of minimalist complex rarely exists. It's almost as complicated as the initiation process, and that is something I'm going to discuss with you next time. So just to sum up here, what I would really hope you have learned today, and then I'm open for questions because we have a few minutes. I really hope that you have an overview of what structural biology can do now, right, with the different methods and the huge steps that we've taken by we as the community, by developing cryo-EM in the recent years, but also by the AlphaFold change of paradigm where you can now predict protein structures with high accuracy. Also these hybrid methods, these methods of integrated structural biology where you can use also cross-linking to make sure your models are correct. So that is a wonderful development because now it's almost unlimited. We can solve very large, very transient, flexible structures of biological assemblies that really occur in the cell, of large assemblies. So that was the first part. And then in the second part, hopefully you could get an idea of what kind of cellular polymerases we have, how these enzymes look like, that they have a highly conserved active site, that actually the chemistry of RNA chain extension, the nucleotide addition cycle, is the same in all living cells, but that when you move to the periphery of the polymerases, they differ. When you move to the factors that they use to find the promoter and to open the DNA, they differ. And this basically periphery, which is different in different enzymes and different in different cells, has evolved to regulate the process, right? Because this periphery allows you to bring polymerases to different sets of genes using different activators that bind to different DNA sites. And also it allows for additional levels of regulation. I briefly touched on this mechanism of DNA opening, which is different for different genes. Some genes require a translocase that consumes ATP to open the DNA. Others can open DNA spontaneously. How do they do it? They do it by the use of binding energy. And I showed you this, highly conserved and positively charged active center cleft of the polymerase that can attract the negatively charged DNA. And that is enough for most systems, bacterial, archaeal, POL1, POL3, and also a subset of POL2 genes to open the DNA. But then there's other POL2 genes that have evolved this more sophisticated opening mechanism, probably also to add a new level of regulation. I've also briefly discussed the point how polymerases find the promoter and the site where they should start transcription. This is, of course, very, very complicated. I could go on for a long time to discuss it. But what you should remember at your stage is that, this textbook pre-initiation complex structure that has now emerged, actually after 20 years of work for yeast and human, it's a highly conserved complex, but it only explains initiation from a subset of genes that actually have a Tata box. And now current research aims at understanding better what happens if you don't have a Tata box. Can the +1 nucleosome maybe be a border element to direct the pre-initiation complex to its promoter? And then, of course, non-coding RNA genes have different upstream sites, like the one I showed you that binds the SNAP-C factor. And then you have a different type of pre-initiation complex. And that is also important to recall. These are the general transcription factors, right? But there's still different subtypes of pre-initiation complexes. Some don't need TF2H for opening. Some will have SNAP-C because they need to be recruited to non-coding RNA promoters. So there's some differences. There's not the one pre-initiation complex. There's a core set of factors, general factors, but the pre-initiation complex can differ on different genes. And then finally, I talked about promoter escape, which means that the polymerase gets phosphorylated by CDK7 on the C-terminal repeat domain. This allows you to destabilize the pre-initiation complex. Initiation factors are more easily dissociated. Actually, also a process that is currently investigated in our lab because it's not fully understood. What is the mechanism to displace those factors? But then the polymerase can escape from the promoter, extend the RNA chain, and form what is called the elongation complex, which contains elongation factors, which are needed for all these different tasks that I briefly described, and which I will go into tomorrow when we talk about the elongation phase of transcription and a little bit more about regulation. But for today, this is what I wanted to tell you. And of course, I'm happy to answer any questions you may have. Thank you very much indeed.